So, good morning. Welcome to the seminar series of the Southern California Environmental Health Sciences Center. We have, I'm Rod McConnell, I direct the center, and we have a special guest today, Valter Longo from our School of Gerontology, who's an expert in the field of aging studies and the determinants of longevity. He has many accomplishments, but among them, he's done pioneering studies on the biology of fasting and of fasting mimicking diets and their effect on physiology and on health, leading to a whole series of trials on looking at various cancers that potentially can be prevented with proper diet, and in particular the role of fasting. He's received numerous prizes, and he's the author of a best-selling book called The Longevity Diet, and hopefully we'll learn a lot today, and I hope you'll give us a few pearls from your book on sort of how we can live to be old.
I'll try. Okay, thanks a lot. Thanks for the invitation. Actually, I'm happy I was happy to come because I've been talking to Doug Finch about pollution, and also Dr. Sayutas for a long time, and so we always thought about collaborating, but that never happened. And so maybe this is the opportunity to do this. But today I'll focus on the. I mean, we don't do much on pollution, and so I try to put together some of the things that maybe are the closest to that, and particularly our work on cell protection and cell sensitization, these differential effects of starvation on normal and cancer cells. And so I started my work in microorganism, yeast, Saccharomyces cerevisiae, and we always go back to this system, and we go back to it because of its simplicity. So this is a unicellular eukaryote, and maybe in 10 years of work that we did on the genetics of aging, we were able to screen. So we were screening actually for stress resistance, meaning that we were looking for what are the genes in this microorganism that make cells more resistant to hydrogen peroxide, more resistant to superoxide, heat, like what we call multi-stress resistant. And and the beauty of yeast, now you can do this with different systems, but back then, yeast, Saccharomyces cerevisiae, was one of the few models for which you could do it. So you could screen and ask the question, what are the mutations that make cells the most resistant and most long-lived, right? So in mutations in this glucose, RAS, adenylate cyclase, PKI pathway, but also, and more so, mutation in this TOR S6-kinase pathway, were the most important in extending lifespan. So yeast that lack RAS2, they live about twice as long as normal, and if they lack these components of these pathways, they live 3 times longer. And there's particularly certain amino acids, serine, threonine, valine, that seem to be very important in accelerating aging in this simple organism. And so if you mutate both the glucose pathways and what we call the amino acid pathway, now you get a 5-fold lifespan extension. And if you mutate both and then you starve it, then you get a 10-fold lifespan extension, right? So the longevity of this simple organism is completely revolutionized by nutrient signaling pathways, but also starvation itself, right? So that means there's still pathways that we don't understand, that we haven't identified, that are driving this, reprogramming into an even longer survival mode. Okay, so then jumping is the only slide on yeast, and then jumping a number of years forward to our work in mammals. And so if you're looking for pathways that are most equivalent to the to those that I just showed you for yeast, is probably the growth hormone, IGF-1, maybe TOR axis, right? So it starts with growth hormone in the pituitary. This growth hormone will be binding to growth hormone receptor everywhere, but particularly in the liver, this growth hormone receptor will activate step 5, which causes the production and release of IGF-1. This IGF-1 is actually the central human growth factor. So if we don't have IGF-1, and I'll show you in a second, the people that don't have IGF-1, they'll be maybe like 3 feet tall. And actually, here they are, right? So these subjects are born with mutations in the growth hormone receptor. And these mice, and the reason, so this is the yeast that I just showed you, the SCH9 mutant, right? Remember the one that lives 3 times longer. It's a dwarf and it lives 3 times longer, but it's also very, very resistant to multiple stresses, right? Lots of toxins, whether it's chemotherapy, hydrogen peroxide, and this is why I'm talking about it here. So this is highly stress resistant. But then you see the flies that have record longevity, they also dwarf, and the mice that have record longevity, they're also dwarf. And these mice and these people have mutations in the same gene, which is the growth hormone receptor, causing a deficiency in the growth hormone receptor and causing this dwarf, causing very low levels of IGF-1 and this dwarf phenotype. And so then just showing you that we can split the lifestyle from the genetics. Of course, both genetics and lifestyle make a big difference. But these people, there's about 100 of these growth hormone receptor deficiencies called LARONs in Ecuador. And with Jaime Guevara, who's an endocrinologist from Quito, Ecuador, we've been studying it for a long time, and they have a terrible lifestyle. So what I'm about to show you has nothing to do with lifestyle or nutrition. It has to do with the genes. And now we know that the little mice that I just showed you, these little mice here, they live 40% longer, and half of them will get to this very long lifespan without any pathologies, right? So it's really remarkable how they can be so long-lived and yet so healthy. So the opposite of what you would think. You always get the comment, well, but if you make people live longer, they're going to be sicker longer. But these mice are exactly the opposite. Right? They're healthier, much longer, right? So, and that's so that's what we were hoping to get in the GHRDs, and that in fact that's what we got, which is the relatives of these little people. They over 20% died of cancer, but cancer deaths are rarely seen in these subjects. And then these subjects are developing obesity at a higher, they have a higher prevalence of obesity compared to the Ecuadorian population, and yet they rarely develop diabetes. We also brought them to Los Angeles, and we did fMRI studies here, and they actually showed a cognitive performance that was more similar to that of younger or much younger people. And so now we just finished a study on cardiovascular disease showing that actually people thought that they were probably going to get higher levels of cardiovascular disease, but it turns out that if anything, they have lower level of cardiovascular disease also, and we're about to publish on that. Okay, so then if you look at so this is the axis that I just told you about: growth hormone, IGF-1, TOR, SCEHSC, and in yeast, where I told you about this part, and in mammals about this part. And if you look at what controls most of this is proteins, right? And specifically certain amino acids within proteins. So then nearly 10 years ago, Morgan Levine, who's now independent, we're working at Altos Lab with Jeff Bezos, a new company. But back then, she was working with Aline Crimmins here at USC. And so we did a study, was asking, what about the Americans that eat high levels of proteins versus the Americans that eat the lowest level of protein? So over 20% of calories coming from proteins versus less than 10% of calories come from proteins. You know, how did they do compared to each other? And it turns out that she came back and said, it makes no difference. The high protein, the mortality in the high protein group and low protein group is exactly the same. It was very surprising to me. I couldn't believe this. So I said, you know, go back and split it into 65 and younger and 65 and older. And I'll show you in a second why we were thinking like that. Before showing the data, I said that at the same time, in fact, this was published eventually in the same issue. The Simpson lab in Australia was also basically giving mice all kinds of combination of food, right, of macro nutrients. So high protein, low carb, low protein, high carb, low protein, high fat. Yeah, and in fact, they concluded that the low protein, high carb diet is the best, right? That yes, the mice eat a little bit more, they gain some adiposity, but metabolically they're healthier and they live the longest. And so then Morgan showed the data. This is overall mortality risk, and this is the low protein, and this is the high protein group, right? So they had a 75% increased risk of dying of all causes, and but they had a 4-fold increased risk of dying of cancer. So why didn't we see anything in the overall population? Because when we asked the 70 and 80-year-olds, those reporting a low protein intake, they were not doing well at all. They were doing worse than those reporting a moderate to high protein intake. So this is turning around based on an age, and at least part of the reason for this is that, and this is true for both overall mortality and cancer mortality risk. And one of the reasons which we suspected is this. So insulin-like growth factor 1, the growth gene that I'm talking about in yeast, flies, mice, in those that had a high protein diet, it was the highest. And this is also suggesting that it's good data, right? So if I'm reporting a high protein diet and I'm 43 or 55, I have high IGF-1 level. And if I report a moderate protein intake, I have a mid IGF-1 level. And if I report low protein intake, I have low or lower. But in the 66 and higher, this does not occur. And so now there is no significant difference whether you have a high protein or low protein. So what may be happening in this group is that you're now driving the IGF-1 to very low levels, and maybe now you're generating malnourishment rather than improvements in IGF-1 levels, which may prevent cancer or accelerated aging. Okay, so then, so what about if we and many others think there is a pro-aging network. Of course, this is a simplification of this pro-aging network, and there's a lot of different things in different pathways in different cell types. But let's just say that for the sake of simplification, we summarize it like that. So proteins driving what I just told you about, and then on the other side, we believe that there's what we call the sugar pathway, just like in yeast, but much more complicated. And now this sugar pathway is affecting insulin, but the sugars are also affecting lots of other genes directly. So there's an indirect effect of sugars and a direct effect, and these are converging on aging and age-related diseases. And so what if you just keep the diet exactly the same and decrease the intake by 25%? And this is probably the most famous anti-aging intervention of all time. It's been studied for over 100 years. It's called calorie restriction. And I would say, and summarize, like all simple intervention, they do a lot of good and they do a lot of bad. And then in the end, you don't get much of an effect, right? So these ideas, that all these solutions are simple. Well, solutions seem to be much more complicated than you think, So yeah, so you can do that. And if you do this in monkeys, and the diabetes or insulin resistance goes from 60% in the animals. And this is a Western-like diet that they use. This is the University of Wisconsin. So 60% of the animal on a Western diet develop diabetes or insulin resistance. And this goes down to 5% in the calorie restricted monkey. So it's just identical diet, but now 25% calorie restricted. And tumors are reduced by 50%, and cardiovascular disease are reduced by 20-30%. So this is reflected by this, right? So this is disease-related mortality, age-related disease-related mortality. So calorie restricted here, and the control monkeys here. But if you look at all-cause mortality, you see how they're much closer together, and so this is indicating that there are trade-offs, right? So yes, they're not getting diabetes, they're not getting less cancer, they're getting less cardiovascular disease, but they're dying of some other causes. For example, infectious diseases, or maybe they do anesthesia and they never wake up from anesthesia, right? So there is a lot of, in a very long lifespan, there's a lot of things that can kill you. And this is probably reflective of this increased sensitivity to maybe a lot of different conditions. Okay, so then Walford was actually my mentor in at UCLA, did the first human study on calorie restriction, and him and other 7 people, they went inside this biosphere 2 environment, sealed environment in the desert of Arizona, and so they stayed in there for 2 years, and in these 2 years they became calorie restricted. So they under the supervision of Roy Walford, who was a medical doctor, they started eating about 25% less, and so actually the monkey study followed this, so I'm showing it the other way around, but just as we've seen for the monkeys, and this is since been repeated by many studies, much larger, randomized, much more careful studies. But here, where they saw systolic blood pressure starts 110 and it drops to 90, glycemia goes from 90 to 70, cholesterol from 185 to 125. So that's really extraordinary effects on risk factors for diseases. But then I showed this picture of Walford, and this is Walford at the end of that period in Biosphere 2, and this is Walford, a year and a half after the exit Biosphere 2. So clearly is being pushed to the limit and probably over the limit, and this probably for the monkeys explains why the overall effects of the calorie restriction seem to be generally neutral. A little bit positive but neutral. Okay, so then we start thinking, well, what can be actually done as effective but more feasible for people, and so lots of different organisms starve for very long periods, right? They completely starve. So the emperor penguins, for example, the males go for a couple of months with no food at all, and they incubate the egg. So they say 2 months, but in some cases they can go as long as 4 months, right? So no food, so they become big, they become overweight, they be obese, and then they use all that fat during these 2 months, and so that they can then incubate the egg. So then the first idea was, what is it possible that we can intervene for just a number of days and starve mice and people, and that these effects will actually last a long time, right? So you starve somebody for 4, 5, 6 days, and then back to the normal diet, And could this be long-lasting effects? And so we first tested this on cancer patient here in USC Norris Cancer Center, and it was it started, I think, around 2010, and it was a water-only fasting chemotherapy clinical trial. And that we thought, of course, cancer patients are going to be very motivated, so they're going to do this, but it turns out that that was not the case. Everybody struggled. I mean, they struggled with it, and the oncologists, I think, they were not very enthusiastic, let's say. So then we went back to the National Cancer Institute, the National Institute on Aging, and we asked for funds to develop a fasting mimicking diet. So it's a low calorie, and eventually we did so, and we were looking for a diet that would change IGF-1, IGF-PP1, glucose and ketone bodies the same way as the water-only fasting, right? So, and what it is a low calorie, low sugar, low protein, high fat, plant-based diet, and that's what we call a fasting mimicking diet. So if you give mice this fasting mimicking diet, and you start this when they're like middle age, it causes a lot of benefits. One of them is that mice, like humans, undergo a age-dependent decrease in white blood cell number. And if you see the mice that have received this fasting mimicking diet cycles for 4 months, or 8 cycles in 4 months, once every 2 weeks, and these are 4-day cycles, the white blood cell count goes back to the young level, and also the lymphoid myeloid ratio, which is a measurement of as mice and humans age, this lymphoid myeloid ratio goes down. You see, this is from a young mouse, middle age, and then older mouse, and now this lymphoid myeloid ratio goes back to not the 4-month-old, but higher than the 16-month-old, right? So you can say that there's the immune system is probably being rejuvenated somewhat by this 8 cycles of FMD. And the rejuvenated immune system is also probably involved in this decrease in all neoplasms in the FMD group, including decrease in lymphomas, and also postponement. A lot of the tumors that are happening later, these are the FMD group, the red squares, and these are the tumors in the control diet group. You see lots of these tumors that are developed here. Now they're here, they're not seen at all. And so suggesting that overall less neoplasms, less lymphomas, and when they occur later. Okay, and then of course, something that we always like to do is the lifespan. And so, like for the monkeys, we want to see this resulting in lifespan changes. And so there's clearly lots of benefits, especially earlier on. Eventually, the very old mice don't like this starvation cycles, and maybe because they have limited fat reserves. And so eventually we have to stop it. And I'll also tell you in a second about the human. And so we tell people after age 70, may not be such a good idea to do these fasting diets. Okay, so then differential. So I mentioned these pathways are involved in multi-stress resistance, and so we started this in the yeast. And so if you take yeast and you starve them, they become resistant to hydrogen peroxide. They become resistant to menadione and lots of different toxins, but also environmental stress. So, for example, heat shock. There's a major increase in the ability to withstand heat. And so then the hypothesis was because the oncogenes, RAS, PKA, etc., are controlling the stress resistance, and in cancer cells, the oncogenes are always turned on. So is there a way to use starvation condition to distinguish all normal cells from all cancer cells? So that now the normal cells, of course, in response to starvation, they'll enter a highly protected multi-stress resistant mode. But because the oncogenes are always turned on in the cancer cells, this is never going to happen, or rarely will happen in a cancer cell. Okay, so that was the idea. And in yeast, this works very well. I mean, this is a yeast that are engineered to be more like cancer cells. So if you're like under with mutations that simulate starvation conditions, and you treat it with toxins, MMS or cyclophosphamide, they're all alive. But if you introduce in the same identical cells, that mimic fasting, you introduce a constitutively active oncogene like RAS-Val19. Now you see that this high resistance and survival goes to high toxicity and death. And so even though we can put them in the same flask, we put them in the same flask. You can specifically only kill the yeast that has this RAS-Val19 gene expressed. Okay, so what about mice? And the experiment was simple. What if you start mice for 48 to 72 hours, and then you give them high dose of chemotherapy? And what happens is that these are the red line are the mice that were pretty fastest starved before chemo, and the gray are the mice that were given a normal diet before the chemo. And you see that the ones that are fasted are all alive, and in some cases, and this is again this works for many different 5-FU, cyclophosphamide, doxorubicin, cisplatin, et cetera, et cetera. So it works for lots of toxins, and that's why I think it's relevant for a group working on pollution and toxins in the diet or in the environment. Okay, so then yeah, so if you see here, the on the left are the mice that are the left are the mice that are oh, I didn't load forget it. You will see much healthier mice on the left compared to the on the right running around, but you won't see that. So okay, so then what are the mechanisms that are involved? And now surprisingly, glucose, which drops by about 72%, I mean, it drops tremendously at 72 hours, and IGF-1 reduction and IGF-BP1 increase were hypothesized to be at the center of these effects. And again, we're looking at these two stress-resistant pathways, the amino acid signaling and the glucose signaling. So the hypothesis was that these are going to be at the center of stress resistance in the mouse as they are in the yeast. Okay, so then these are mice that are exposed to doxorubicin, and again, they're fasted, they're all alive, they're a normal diet, all that. But then if they're fasted and we inject insulin into them, you see that we don't completely reverse their resistance, but maybe we eliminate half of the resistance. So now IGF-1 alone is responsible for reversing lots of the protection of these mice against chemotherapy. And the other side is glucose. And here, using dexamethasone or rapamycin, we can actually increase glucose level. And if we increase glucose level and then we expose the mice to doxorubicin, you see that they're sensitized to doxorubicin, and they're sensitized in both cases, they're more sensitive. Now, interesting here, because rapamycin is actually an anti-aging drug, but yet because it causes hyperglycemia, is making cells more sensitive to the chemo. And so insulin is sufficient to reverse this toxicity, confirming the role of glucose. And actually, if we starve these mice, even though they have rapamycin or they have dexamethasone, and by the way, both of these are given frequently to cancer patients in the clinic. But if we use the combination of fasting conditions with the dexamethasone or rapamycin, now we can completely reverse this toxicity. So this is how powerful the fasting conditions are in blocking the toxicity of these chemotherapy drugs in normal tissues. Okay, so then Chao Wei in the lab actually back then was looking at cyclophosphamide toxicity. And so when you give cyclophosphamide to mice, whether the PF means prolonged fasting, cyclophosphamide plus or minus prolonged fasting. So if mice were exposed to cyclophosphamide, whether they were fasted or not, it didn't matter. The white blood cell count, the lymphocyte count, and the white blood cell count would drop, right? So the starvation conditions were not protecting the white blood cells from chemotherapy toxicity. But a very interesting thing she saw was that after 6 cycles, the level of the lymphocytes went back to normal only in the fasted group, right? So, and this is hard to explain with protection. So she started looking at, could it be that there is regeneration involved? And so she started looking at this, and then she demonstrated this regeneration by either having a bone marrow transplant from a donor that has been treated with cyclophosphamide, or a donor that has been treated with fasting plus cyclophosphamide, and irradiating, and transplanting into a irradiated mouse, and then looking at peripheral blood cells and lymphoid myeloid ratio, and then the composition of the presence of stem cells in the bone marrow. And so you see here, eventually, the bone marrow cells are able, from the starved mice, are able to produce more peripheral blood cells, are able to restore the lymphoid myeloid ratio more effectively, evidence of more functional immune system. And they're doing so probably by having more stem cells, LPHSCs, they're self-renewing hematopoietic stem cells, and also the other stem and progenitor cells, so much higher if the bone marrow came from these fasted mice versus the non-fasted mice. And also rejuvenation, right? So these are all mice that received prolonged fasting cycles. You see that in the old mice, the lymphocyte portion goes down. This is young mice, and lymphocyte portion goes down in the old mice, and the myeloid portion goes way up. And these are actually portion of stem cells that are either lymphocyte biased or myeloid cell biased, right? So they're biased to generate this particular cell type, and so you see here that the FMD is able to restore a more youthful profile of stem cells that are higher in lymphocytes and lower in myeloid cells. Okay, so then the starvation conditions are causing, through the growth hormone IGF-1 downregulation, but also through, I don't have time to talk about it, through the downregulation of PKA, which again is the glucose signaling gene. So again, growth genes and glucose signaling are now able to not only protect, but also cause a reprogramming of the stem cells into a more regenerative mode. Okay, so then this is the summary of what we call differential stress resistance. So the protection of normal but not cancer cells. Normal cells are protected while IGF-1 goes down, glucose goes down, but then the proto-oncogenic pathways are also downregulated, and this leads to activation of stress-resistant transcription factors and cellular protection and low growth. And tumor cells rebel. It doesn't really matter which oncogenic mutation is present. Any oncogenic mutation is going to prevent this induction of protection, and it's going to keep the cancer cells growing under low protection. Okay. So what about then as we were approaching clinicians here at USC Norris, they were asking, well, how do you know you're not protecting the cancer cell, right? So if you're protecting even slightly the cancer cell, now we got a problem, because you cannot expect that we're gonna, if this is successful, that we're gonna revolutionize the cancer treatment for this. So you have to demonstrate no protection of cancer cells. And again, we started with yeast and looking at a differential, what we call differential stress sensitization. So that if you stress with heat shock, hydrogen peroxide, menadione produces superoxide and hydrogen peroxide. If you stress the cells with these factors, it doesn't really, if there's a lot of glucose, if there's excess nutrients, they don't really care that much. Maybe a little bit hydrogen peroxide, but in general, it doesn't really matter whether they're wild type or they're cancer-like. But if you starve them now, you see that how the RAS, the constitutive activity of the oncogene is now generating this differential sensitization. So in some cases, you're looking at hundreds of folds of difference between resistance to hydrogen peroxide. These are serial dilutions. So resistance to hydrogen peroxide is still very high in the wild type, but is gone in the cancer-like yeast. So and this is again the FMD. So what if you apply either lung cancer model or breast cancer model, and you look at cancer-free survival? What portion of the mice can you cure? Right? Not delay growth, but actually go after every single cancer cell and cure them, or cause a long-term regression. And so if you look at lung cancer, chemo, zero cancer-free survival, fasting, zero cancer-free survival. If you use a low level of this triple negative breast cancer, you get about 20% chemo, 20% with fasting. But what I wanted to show, of course, is the combination, right? So now you have fasting or fasting mimicking diet plus the chemo, and this is actually FMD plus the chemo. And now you see 40 to 60, in lots of different mouse models, you see 40 to 60% cancer-free survival. So this combination is able to sort of search and destroy cancer cells no matter where they are.
This is animals, I'm sorry, this is animals who already have cancer?
This is animals that are injected with the cancers, with cancer. And 100% of which will die of cancer if untreated. Yeah. Or if treated with chemo or fasting. Right? If you just give them fasting cycle, they all die of cancer. So then we started thinking about, really lots of patients don't die of cancer. Lots of cancer patients don't die of cancer, they die of the treatment, right? And so we started thinking, well, wouldn't it be interesting if we could find like completely non-toxic intervention that are as effective as, or maybe more effective than the toxic ones like chemo. And so we knew from the work with Lou Cantley, the East Coast, that vitamin C was very effective in treating KRAS mutated and BRAF mutated colorectal cancers, and through a reactive oxygen species dependent mechanism. And so this is actually Myra in my lab in Milan. So Myra started looking at the same thing in using lots of different cancer lines, 3 colorectal, lung cancer, pancreatic cancers. And sure enough, the vitamin C, so this is fasting, this is vitamin C. So the vitamin C is working, as Cantley had proposed, is working for these KRAS mutated cancers, particularly for the pancreatic. But then when you combine fasting with vitamin C, now you see this big effect of the combination in killing these KRAS mutated, but also the BRAF mutated cancer cells. And so in looking at, well, this is in vivo, you see again now this fasting mimicking diet plus vitamin C, highly effective, and I'm not showing it, but more effective than the chemo, right? So it's more effective to have 2 anti-aging interventions against, and these KRAS mutated tumors are about 25%, I think, or so, something like that, of all cancers, right? So KRAS and BRAF. So this is working very well. This combination, even though to normal cells is completely non-toxic, and this is the that was the human cancer, and this is the actually mouse version of this of colorectal cancer. So it's working, independently of the line that we used. And it's working by causing a high level of DNA damage. Only, you see that these three lanes are fasting alone or vitamin C alone, but the damage, the DNA breaks are occurring only when we combine fasting with vitamin C. And why is that happening? Well, we had published on something we call anti-Warburg effect. And so if you starve at least in the colorectal mouse model, if we starve the mice, then the colorectal cancer are going. So normally in cancer, you have a Warburg effect, and lots of cancer turn to glycolytic source of energy, but not just energy, also biosynthetic intermediates. And so, and the starvation that is forcing the cancer cells to return to oxidative phosphorylation, right? So that's why we call it anti-Warburg effect. And not surprisingly now, they're producing a lot, in a synergistic way, they're producing a lot of free radical, a lot of superoxide, hydrogen peroxide, etc. Only when we combine fasting and vitamin C. And they also produce a lot of free iron, which is not seen in the vitamin C or fasting alone. And the reason for this free iron is, and this is what happened, we think explains the difference between vitamin C alone and vitamin C plus fasting, is that in the vitamin C alone, ferritin is induced, right? So, and this is in vitro, in vivo, you still see the same effect. So there's a high induction of ferritin, which actually scavenges iron 2. So it makes it unavailable to create free radicals. And the fasting is reversing this induction of ferritin level. So now ferritin is back to the normal level, and this probably justifies this free iron, this availability of free iron. And what controls ferritin? It turns out that we already had shown induction of heme oxygenase in other tumors, and we've shown that fasting reduces the heme oxygenase 1. And so we think because heme oxygenase controls ferritin levels, we think that well, actually Meyer demonstrated that that's what it was. And so then the summary is that normally vitamin C can cause some free radical production, but most of the iron is scavenged by the high heme oxygenase 1, high ferritin level. As this is reduced and the ferritin reduced, a lot more iron 2 available, and this iron 2 can react with hydrogen peroxide and generate hydroxyl radical, and that's probably what is doing a lot of the damage in the cancer cells and a lot of the breaks. Okay, so what about clinical trials? Well, started here at USC with Tanya Dorf and David Quinn doing the first pilot study with fasting water only 24, 48, 72 hours, and starting already seeing some initial evidence of protection. But this was very preliminary, and then eventually University of Leiden looked at the erythrocytes and thrombocytes and showed that there was the patients that were fasted in combination with chemotherapy, they showed a protection of the erythrocytes and protection of the thrombocytes from chemotherapy. And then the Mikkelsen lab in Charité Hospital in Germany did a study with breast and ovarian cancer women receiving chemotherapy and showed that the quality of life did not, these are multiple cycles of fasting, of chemotherapy, C1, C2, C3. And so there was no difference in quality of life in those the women that fasted, but there was a significant difference in quality of life decrease in the women that did not fast in combination with chemotherapy. But the larger study is this 125 patient HER2 negative stage 2-3 breast cancer study. And this was done at the University of Leiden in 12 different centers. And actually, the compliance was relatively low. And in spite of low, meaning that women that did they received the chemo, 6 to 8 cycles of chemo, either with or without the fasting mimicking diet. And even though there was a relatively low compliance, this still worked, and the clinical response, the non-responders went from 27% in the regular diet group to 11% in the FMD group, and this was a significant difference. But if you look at per protocol, meaning if you actually look at the women that completed the FMD, all the cycles of the FMD, or most of them, then the difference went from 27% to 5%. So now there's a 5-fold difference in non-responders. And if you look at the pathology, you also see a dose response in the pathology. So the women that were compliant to none of the cycles of the fasting mimicking diet were much less likely, so less than 10% were 90 to 100% cancer-free. So this is looking at the Miller pane is looking at the activity of cancer cells within the masses that were removed from the breast of these patients. And so, so now it goes from over 50% to less than 10%. So the pathology and the clinical response are matching, suggesting that this is probably working pretty well. Okay, then another trial in the University of Genova and the Italian National Cancer Institute looked at actually the effects of these on weight and hand grip strength, muscle function, muscle mass, and fat mass, and showed no difference of many cycles of the fasting mimicking diet in hand grip. Of course, the criticism was a cancer patient cannot undergo many cycles of this FMD, but that doesn't seem to be the case. They can do it, keep maintaining the strength. Actually, the phase angle, the measure of muscle function, went up instead of down, and the fat-free mass also went up. And this is in agreement with some of the work that we're doing, showing that this may be contributing to regeneration, stem cell activation, and regeneration, potentially also in the muscle. But just like the mice, IGF-1, leptin, C-peptides are down, and ketone bodies are up. And some of these effects last for weeks, so they're not temporary. For example, IGF-1 and leptin, the effects on these two are long-lasting. Okay, so then why does it work? Well, it revolutionizes the environment both for the normal cells and for the cancer cells. And for the normal cells, these changes in glucose, IGF-1, insulin, leptin, etc., seem to be beneficial. They're protective. But for the cancer cells, these changes cause probably many cell types this anti-Warburg effect, relying much more on mitochondria, but the mitochondria, most cancer cells are damaged, in some cases irreversibly damaged. So this anti-Warburg effect is causing lots of problems, reactive Austrian species. And this is probably what makes this, I didn't talk about the immunity component of our work, but is making these cancer cells much more visible to the immune system. And now we see immunosuppressive macrophages going down and T cells going up, or at least becoming more aggressively attacking the cancer. And so we see in mice and in humans, we see a higher infiltration of the T cells in the tumor of patients and mice that have been treated with the fasting mimicking diet. And this is all I have. And I just, I think I thank the people, but most of the cancer work it was done by Charlie Chang, Roberta Bono, Valentina Villani, Sebastian Branders. Thanks. Questions? Yes.
I remember when we were measuring the IGF-BP1 in mice for one of the experiments, there was the 11-fold increase. And you mentioned that you thought that might be what is driving it. But then in just a few slides back, the IGF-BP1 changes were significant after 3 weeks in humans. Do you know?
Yeah, well, BP1 I didn't mean that BP1 is driving it, but the IGF-1, low IGF-1 and high BP1. So you have low levels of IGF-1 and high level of an inhibitor of IGF-1, right? So yeah, so and now keep in mind that we're looking at effects in the 5 days. Most of the anti-cancer effects happen in the 5 days. So that's what we're focusing on. So we believe that the 5 days is enough to kill a lot of cancer cells, right? And so then the 3-week effect is probably not necessary. I mean, it may even be detrimental, right? So eventually, so for example, there's a paper that I just reviewed that was suggesting that, I think it was testosterone, that the immune system, it's not published, but that the immune system actually needed higher level. So the testosterone will go down temporarily during this starvation, like estrogen. But then they were showing that the testosterone high was necessary for the activation of the immune system, right? So what does it mean? That it means that certain things happen during the fasting and the fasting making diet, but then those are followed by in the refeeding period, they're followed by other things, which could be as important, right? So you're pushing the system from one side to the opposite. And that's probably why it works so well. And some of it is it puts you in a bad state, but then I activate the immune system, and IGF-1 might actually be good to activate the immune system. And so it's testosterone, and maybe BP1 should be low. And this could also explain maybe why the pharmaceuticals, 7 companies had developed IGF-1 receptor blockade, and they all failed because they were permanently feeding patients with, injecting patients with IGF-1R receptor blockers, which may not be a good idea, right? Because again, the immune system and lots of other systems are depending on growth factors.
So do you think the IGF-BP1 might have been elevated in like the first measurement that you did? I forget what time after treatment it is.
Yeah, usually temporarily is way up, right? As I showed earlier, in both people and mice, but then eventually comes back down. But even if IGF-BP1 was not significantly higher, as long as IGF-1 is lower, there is probably a change in the IGF-1 signaling that is sufficient to get some of these effects, right? Now, of course, you know, what will happen if patient fasted for longer? I mean, now we have to sort of do much more, like thousands of patients with this. If once that is successful, so for example, with some blood cancers, myeloma, multiple myeloma, CLL, some patients have done much longer and they see very beneficial effects. And those are definitely going to drive IGF-1 lower and BP1 higher.
So how much variability do you see in the response to these, periodic testing?
Variability in which model?
Kind of in humans. And in kind of any, let me rephrase it. What's the outcome you see the most variability in humans?
We will have to look at the data. Now we've already tested hundreds of patients, so different responses. It also depends on the baseline, right? So if you start with a normal blood pressure, I didn't show those data, but if you start with a normal blood pressure, unlike what we see for calorie restriction, the blood pressure doesn't decrease further. But if you start with hypertension, and then we see a steeper decline, right? And this is true for C-reactive protein, IGF-1, cholesterol. So almost everything that those that start the worst do the best. And that's not necessarily due to weight loss, right? For example, we see biological age decrease, and that's independent of weight loss. So that those that lose more weight are not necessarily reducing their biological age the most.
I have a question. So, because you're studying longevity, what's the impact of fasting mimicking diets in genetic clocks?
Yeah, so we did that in collaboration with Steve Orvat. and it decreases it. But I also learned there's a lot of clocks, and there are a lot of clocks that orbit uses, and in different clocks, we saw different rates. But in general, we saw FMD cycles in the mice. We haven't done humans yet. Well, we're doing it right now in southern Italy, but we don't have the results yet. But in the mice, yeah, in most of the clocks, we saw a reduction in epigenetic age, but not all the clocks. Sorry.
Okay, I think the growth hormone plays a critical role in the early life when people are developing. How do you see the role of early life development in longevity determining that?
It's a very good question. Yeah, so there's a lot. I was just yesterday at a conference, and Andy Dillon was showing the work in worms, and so in worms, an exposure to, let's say, mitochondrial damage early in life, it can completely change the survival. And also, like in mice, methionine restriction or protein restriction early in life, even though you remove it, and you put back methionine and then you feed it normally, it can affect the entire lifespan, right? So yeah, there's definitely a developmental wiring effect, but then you can intervene, for example, with calorie restriction, FMD. So the FMD thing that I showed you was started at 16 months, and it still works very well. And rapamycin was started at 20 months, and it still works very well, right? So yeah, there's probably both, right? You can have a developmental effect, and you can have a post-developmental effect.
I was just going to ask, you said, for folks who are doing environmental epidemiology or toxicology, cyclophosphamide or dexamethasone, those are potent toxins, potentially an analog to an environmental exposure or model for an environmental exposure. So
and this is why I picked this, because I don't work on environmental exposure, but I thought that, and we're also doing work on repair, right? And we haven't published a lot of the work on repair, but there is a protection effect, and there's also repair effect. Like I showed, remember, with the stem cells now, the damaged white blood cells, and then the stem cells eventually replacing that, right? So there's an opportunity both in protection and in repair of the environmental damage.
And so, but those are quite different poisons.
Which ones?
Cyclophosphamide and dexamethasone and rapamycin. Is there a common mechanism for?
Well, but dexamethasone and rapamycin, we were just using it these are not toxins, right? Dexamethasone and particularly rapamycin is an anti-aging drug, right? It blocks TOR, but it just happened to cause high glycemia. And so we were just interested in hyperglycemia's effect on cell sensitization, right? The toxins were doxorubicin, cyclophosphamide, 5-FU, MMS, alkylating agents we've used, and it usually works for most of them.
Are those? I guess the point is, what's an environmental analog of those toxins?
Well, for example, paraquat, right? Paraquat exposure, paraquat, this FMD protects very well, and it causes a repair of paraquat toxicity, right?
Oh, that's interesting.
Paraquat, menadione. In fact, I showed you menadione. Paraquat and menadione are very similar. So paraquat used to be sprayed in the fields of, probably still sprayed somewhere.
Everywhere.
So there's, that's one example. But the beauty is that it really doesn't matter what the toxin is, right? It's just fundamental multi-stress resistance. So because yeast and bacteria have to be protected against anything that maybe the other bacteria are producing, right? So they became resistant to lots and lots of different chemicals. And this is why even HCEHSC, remember I showed HCEHSC and menadione and chemotherapy, and it just keeps on working, So if you take a highly protected yeast, it's hard to find one thing that they're not protected against, because they're G0, they're really generating a shield, both physical shield and molecular shield. And then there are also probably in the mammalian cells, you also probably have, drug breakdown pathways being activated. We haven't looked at that, but that's probably also part of the protective system.
So paraquat, you fast them, then you dose them with paraquat and they do better?
Yeah, you fast them and then you dose them with paraquat, or let's say dobutamine or paraquat. You treat, you do you cause cardiac toxicity, and then you start the fasting Mimicking diet cycles, and the heart becomes more resilient, right? So it's also, you can do it after, right?
Really?
So yeah, and probably because it's activating autophagy, mitophagy, and so probably there is an opportunity to help the cell get rid of damage, right? And yeah, in some of it may be regenerative in nature. So you replace an old cell and you put in a new cell, which is still, good news for any environmental toxin. But part of it is probably just doing it without replacing the cell. Yes.
I have a question about your findings in high versus low protein diets. I'm wondering if you guys looked at different sources, like animal versus plant sources of the protein, and if not, do you think it's driven maybe by animal sources?
Yeah, so we looked at that. And in fact, it was in the paper. And if you look at overall mortality, the people in America, not necessarily American, the people in America, they were having a high protein diet, but it was mostly plant-based, did not have an increase in overall mortality, but they still had an increase in cancer mortality. And it went from fourfold, I think, to threefold increased risk. And then Giovannucci Harvard published something very similar. I was also part of the paper, and again concluded that high protein is clearly detrimental. High protein, high animal protein, high animal fat. But if it's high animal protein and fats from plant-based sources, then he actually claims a reduction in the overall problems. Right now, he hasn't looked at. He looked at cancer, but in those papers, no, he also looked at cancer, but there's a little bit of a split. But in general, we agree that the plant-based proteins are better, but he feels that they're actually even better than to have a high protein plant-based is the best of all. That's what he claims. We don't probably think so. Also because it's very difficult to have a high protein diet if you're plant-based. You have to eat, kilograms of legumes every day, right? And so I think if you look at this high protein plant-based group, is relatively low protein compared to the animal group, which is, maybe have 20% or 25% protein. And so it's very easy if you eat steaks and chicken to get to that level. Yes.
Just following up on that, actually, is there any, I think the animal industry, uses a lot of growth health in packing up their animals. And so is there any role for that additional growth hormone to have to, so is there a difference in organic meat versus non-organic?
Yeah. Believe it or not, we spend a lot of government money to look at exactly that. Sure that we were gonna see the organic do much better. And we did the entire lifespan, extremely expensive study, right? Absolutely nothing, yeah. We were shocked, because, and we like McDonald's, we went and got the worst of the worst. And we thought, but as long as it was animal proteins and as long as it was matched, the lines overlap perfectly, right? So yeah, we were very disappointed, We were trying to prove a point, which was not provable. That doesn't mean that these are mice, right? So it could be that mice are much more resilient in general, and it doesn't make much of a difference. But that tells us, I think, that the amino acids, particularly content of the animal proteins, is driving the IGF-1 levels, is driving the insulin levels, the growth hormone levels, and so it's driving the accelerated aging and also this differential protection.
I guess totally separate question is, I think there are, there are conditions in which children might be given birth hormone because they're, not growing or what have you. Has anybody ever looked at that repercussion? Ancillary repercussions too?
Yeah. Yeah, they looked at it and they don't see much. They see some increases in some cancers, but it may be that the high growth hormone is understood by a child, right? So that meaning that when you're that young, the high growth hormone is part of that because IGF-1 and growth hormone is very high in very young people. So that's part of. So we have evolved probably a very good system to protect against even extremely high levels of growth hormone as long as we're young. What we haven't developed is probably to do the same when that happens later in life. And now that we have people eating 2-3 times more proteins than they should. So for example, colorectal cancer is moving to earlier ages, right? So is it possible? It's because we're loaded with proteins now, right? So some of the cancers may be much more sensitive to these growth factors. And, so why are these cancers all of a sudden moving to younger people? So that this sort of protein craze, every other commercial on TV is about protein. And a survey shows that people believe the healthiest food for you is proteins. And we will argue that that's absolutely the worst food for you for health and longevity. But in a survey, people will say, we'll put proteins as one of the healthiest foods that you can, or ingredients that you can eat. Yeah.
Brittany? Any thoughts about, there have been a series of studies looking at timing of eating.
Yeah, time restricted eating, it's called, yeah.
Yeah.
Yeah, I was there with, in fact, Sachin Panda yesterday was also there in San Diego presenting. And so Sachin is the one that's done many clinical trials and basic research on time restricted eating. Yeah, so TRE is probably, it's every day, right? And that everyday restriction on the time of consumption of food is having clear effects on metabolism, on sleep, and lots of other things. So it's very different because I think the longer fasting mimicking is causing, effects on stem cells, the shrinking organs. It's one thing that I haven't shown, but Ansel Keys back in the 60s had done these semi-starvation studies in humans. And after, I think it was 24 weeks of this severe calorie restriction, they showed that the heart dimension was reduced by 41%. So now the human heart became 41% smaller. And then 20 weeks later, it went back to the normal size, right? So, so then there is much stronger effect of the long starvation. But then again, if you do something every day, that accumulates, right? So then, we think that both of them are important for very different reasons. So now Sachin also showed that people in the US on average eat for like almost 15 hours a day. And so we're eating for longer and longer periods, and this is probably interfering with sleep. And so when he reduces that from 15 to 12 or 11, people get a lot better. He just published one study with the firemen in the San Diego area and showing lots of improvements there.
Thanks for coming.
Thank you. Thank you.
Really interesting presentation.
So for those of you who are interested in continuing the discussion, we have put up there in room to the video.
And everybody was doing it. Good. If you are.
I just said that, yes, that there is, after the log on the fasting.
But that's not what we're to do in the nation. We need to have some. You're worried about it. You just got paid already. I mean, like, but it's all, it's not really, between the life of the student and Next one over there. 1 minutes.
Yeah.
Oh, yeah. Oh. my goodness. Okay. Yeah. This one went up. Yeah, but it is like everything. That's quite the long time. No, girl. You don't want it. But in person. I don't know, it's very different. Yeah. It's been rough. It's been real rough. But how much did you spend it? Yeah. We'll have to, reading one of two. Exactly. I'm not being tired of it. It's a white chart. Nice, thank you for that. I don't need to bother. I'm just looking for a charger. No problem. Is anyone good? Can anyone help with that? I think these are really good. No. That I had helped get some. Yes, but I think she's gonna come back.
How do you feel for like?
Which
Can you vote? Yes, there you go. Yeah, just. No, I actually caffeinated. I. Yeah, my cat was just like up all night. And so he was like, he wanted to get so black. He's like crawling all over my bed, like trying to get me my laptop.
There was like a seminar out here, and we were like anxious to go, so we got the whole part.
Also, it's kind of weird.
Yeah, I was supposed to. Me too. Yeah, actually closed up. No.
Which you want, but. Do this and go home.
Let's get to be short. Hey, what do I do with this?
Just change the source, maybe?
Maybe.
Two laptops.
Okay.
And then. I think so, sometimes, lectures,
Perfect.
then, and we can wait to go back.
Yeah.
Like a formality.
This is not yours? That's from previous? Actually, they're.
In case you need